What if we could edit our genes to cure genetic disorders? Imagine having the power to rewrite the code of life, to correct errors that cause diseases, and to unlock new possibilities for human health. This is the promise of gene editing, a field of study that has been revolutionized by the discovery of CRISPR-Cas9. This technology, derived from a defense mechanism in bacteria, is like a pair of molecular scissors that can cut and paste DNA with unprecedented precision. However, despite its potential, CRISPR-Cas9 is not without its challenges. Its size, for instance, makes it difficult to deliver into cells, and its activity sometimes leads to unintended edits. These hurdles have sparked a global race to find better, more efficient gene editing tools. But what if we told you that a new player has entered the field, and it's smaller, more compact, and potentially more efficient? Meet CoCas9, the new CRISPR enzyme discovered by researchers from the University of Trento. This compact enzyme, with just over 1,000 amino acids, was extracted from an uncultivated Collinsella species, a bacterium found in the human gut microbiome. In the ever-expanding universe of gene editing, CoCas9 is a fresh player, bringing with it the potential to expand the use of gene editing for therapeutic applications. To find CoCas9, the researchers embarked on an extensive analysis of metagenome-assembled genomes. These genomes were primarily extracted from human microbiomes, the diverse community of microorganisms living in our bodies. By delving into this microscopic world, the researchers were able to uncover a treasure trove of type II CRISPR-Cas loci, the genetic locations where CRISPR-associated proteins like Cas9 are found. Among this vast variety, CoCas9 emerged as a standout. It's a highly active and precise nuclease, an enzyme that can cleave the strands of DNA and RNA. But what makes CoCas9 truly unique is its reduced molecular size. This compactness is not just a fascinating quirk of biology, but a feature with practical implications. When it comes to gene editing, size matters. The smaller the enzyme, the easier it is to package with guide RNA and deliver into cells. CoCas9's small size also makes it suitable for packaging within adeno-associated virus vectors. These vectors are commonly used to deliver genetic material into cells during gene therapy. Therefore, the discovery of CoCas9 could open up new possibilities for in vivo gene editing, where changes are made directly in the cells of a living organism. Experiments in human hematopoietic stem/progenitor cells and murine retinas have already demonstrated the potential of CoCas9. In murine retinas, CoCas9 achieved up to 35.6% editing efficiency when targeting the RHO gene, which is mutated in a common form of autosomal dominant retinitis pigmentosa. With its high precision and reduced molecular size, CoCas9 stands out as a promising tool for gene editing. It's a testament to the power of exploring the natural world, even at its smallest scales, to find solutions for some of our biggest challenges. So, how does this compact enzyme perform when put to the test? Imagine a stage set for a grand performance. The spotlight is on CoCas9, the newly discovered CRISPR enzyme. The initial act? An experiment on human hematopoietic stem and progenitor cells. These cells are the building blocks of all other blood cells, making them essential for the production of blood and immune cells. And guess what? CoCas9 showed promising results, demonstrating its potential for gene editing applications even in this complex cellular environment. The second act of our performance takes us to a different setting, the murine retinas. Now you might be thinking, why murine retinas? Well, the retina is a layer of tissue at the back of the eye that converts light into neural signals and sends them to the brain for visual recognition. The reason for this choice is to test CoCas9's ability in a real-world, medically relevant situation. You see, a common form of autosomal dominant retinitis pigmentosa, a type of progressive retinal degeneration, is caused by mutations in the RHO gene. And this is where CoCas9 takes the stage once again. The results? Nothing short of impressive. CRISPR-Cas9 achieved an editing efficiency of up to 35.6% when targeting the Rho gene. This means that over a third of the targeted genes were successfully edited, demonstrating the enzyme's high precision and efficiency. But the performance doesn't end there. All this is done within the confines of its compact size, which allows it to be packaged with guide RNA within the tight quarters afforded by adeno-associated virus vectors. This enables in vivo gene editing for therapeutic applications, further highlighting CRISPR-Cas9's potential in the field of genomic research. The results are clear. CRISPR-Cas9 has immense potential for gene editing applications. It's like a new star being born in the galaxy of genomic research, shining a light on the possibilities for the development of therapies for genetic disorders. And we can't wait to see where this star's journey takes us next. But why does this matter? What does CRISPR-Cas9 bring to the table? Well, let's dive into the significance of CRISPR-Cas9. First off, CRISPR-Cas9's size is a game changer. This compact enzyme, with just over 1,000 amino acids, is perfect for packaging within adeno-associated virus vectors. These vectors are like tiny delivery trucks, transporting the gene editing tools to the right spot in our body. But the cargo hold is limited. The smaller the package, the easier it fits. So with CRISPR-Cas9, we can now carry out in vivo gene editing for therapeutic applications more efficiently. But the magic of CRISPR-Cas9 doesn't stop at its size. This enzyme is naturally evolved, which gives it an edge over modified enzymes. It's like comparing a custom-made suit to an off-the-rack one. The custom-made one, in this case CRISPR-Cas9, is tailored to our needs, expanding our CRISPR toolbox to develop therapies for genetic diseases. And let's not forget the importance of collaboration in this discovery. The University of Trento's molecular virology and computational metagenomics laboratories joined forces, unlocking a vast natural reserve of CRISPR-Cas9 systems for human genome editing. This partnership proves that when different fields unite, we can push the boundaries of science even further. CRISPR-Cas9 is more than just a new enzyme. It's a significant step forward in genomic research and the development of therapies for genetic disorders. To wrap things up, the discovery of CRISPR-Cas9 has ushered in a new era in the field of genomic research and the development of therapies for genetic disorders. This compact, highly efficient enzyme found in our very own gut microbiome is indeed a game changer. Its potential for in vivo gene editing and its suitability for further engineering optimization make it a powerful tool in our CRISPR toolbox. With CRISPR-Cas9, the future of gene editing and curing genetic disorders looks brighter and more promising than ever before.